about pigs, really. I also have to tell you that I'm not an animal science people. I am a laboratory people. In fact, I am more a biochemist than anything else. So our work has been always done in collaboration with people that knows about animals and animal welfare. So, well, let's start now with stress and pigs. And in fact, fattening pigs are really subjected with too many types of stress, going from housing, types of housing and management, transport to the slaughterhouse, and also in the slaughterhouse there is mixing of the animals, there is noise, there is full pens, etc. And all these steps they may become really stressful factors for pigs. We have worked also in the positive way to affect welfare, that is on environmental enrichment. So what I'm going to present you today is first our work on environmental enrichment in pigs. Second, I would like to present part of our work in intrauterine growth restriction. That is, maybe it's not really like a welfare subject, but it's also related with the well-being of the animals. And third, if I have time, I would like to present also another work on pig housing and pig management, but there is not much neurobiology on this work, so I will just let it just in case we have time. So let's start with stress biomarkers. We do have several possibilities to manage and to try to quantify in an objective way stress using plasma or serum markers. Among them, there are, of course, the stress hormones, cortisol, catecholamines. Cortisol mainly affects also metabolism, so it leads to lipid mobilization. So, for example, cholesterol is often increased in stressed animals. On the other side, stress also affects oxidative stress. So do we have oxidation markers? We can measure antioxidant enzymes in serum and plasma. And finally, we have an involvement of the innate immune system that leads to the production of cytokines. And then these cytokines act mainly on the liver, and the liver produce acute phase proteins that can be also determined on the plasma or serum samples. These acute phase proteins are markers of inflammatory or infectious conditions, but can be also altered by stress. But of course, we are interested now in the brain. The brain in the pig has the advantage to be relatively large. That means that we have a kind of material to make several kinds of studies and perform several types of experimental work. And from the point of view of the pig as an animal model, it's also similar to the human brain, much more similar than the brain from rodents. For example, the pig brain has convolutions like the human one, whereas the brain from mice or rat is what is called lissencephalic, and doesn't have convolutions. So what we have done is to dissect several areas in the brain that are related to some kinds of moods or stress or several kinds of reactions of the organism at the neural level. So we have worked with the hypothalamus that is here, with the prefrontal cortex. We have also work in the hippocampus, is this here, and also the amygdala here. And although I don't have here the picture, we have sometimes also worked with the striatum. Then now we are talking about neurotransmitters, and the ones that we have been working most are those from the what is called monoaminergic system. There are two types of neurotransmitters. The first one are the catecholamines that are mostly norepinephrine or noradrenaline, no? Depending on the country, and also dopamine. This is the catechol ring, so those are catecholamines. And then we have serotonin, which is the other main neurotransmitter that is an indolamine. This is the indole ring. And the other name for serotonin is 5-hydroxytryptamine, 5-HT. And those are the most important neurotransmitters for us. Of course, there are others. There are others like glutamate or GABA, GABA. But we have worked mostly on these monoaminergic neurotransmitters. These pathways, these neurotransmitters are related to several conditions, as I told before. No, norepinephrine is mostly related to stress, dopamine to reward, to pleasure, but it has complex functions. Serotonin is mostly related to mood, to memory processing, and those are the brain areas that we have worked on. Amygdala is related mostly to emotion and fear, to mood in general. The prefrontal cortex is related to these higher abilities of the brain, cognitive behavior, decision-making, social interactions. The hippocampus has an important role in memory and also in spatial coding. The striatum in reward and addiction. And the hypothalamus is related mainly to the neuroendocrine signaling and hormonal signaling to the rest of the organism as the origin of this axis. No? So first of all, I would like to present how these neurotransmitters are synthesized and degraded, because we are going also to determine some of these intermediate metabolites. So dopamine is here, and dopamine is the precursor of norepinephrine, and both they come from the amino acid tyrosine. This is the enzyme that regulates the pathway, tyrosine hydroxylase, that joins this hydroxyl group in the ring. And this is the way how dopamine is degraded by these two enzymes, monoamino oxidase and COMT, and those are the metabolites that dopamine is converted into these metabolites. The most more relevant or more abundant are DOPAC and also homovanillic acid HBA. And referring to serotonin, serotonin comes from another amino acid that is tryptophan. Tryptophan is converted in serotonin also by tryptophan hydroxylase, which is again the regulatory enzyme of the pathway. And serotonin is degraded also via monoamino oxidase to this metabolite 5-hydroxyindolacetic 5-HIAL. So we can separate and identify and quantify all these molecules by using a liquid chromatography system, an HPLC system that is conducted on what is called reverse phase, and using an electrochemical detector that puts a filter in a way that the peaks, the molecules that are detected are those. And this is a typical chromatogram with all the peaks. We can identify and quantify also, because it is a quantitative technique if we use an adequate set of calibrators. And we can identify and quantify noradrenaline, norepinephrine, dopamine, DOPAC, HBA, serotonin, and 5-HIAA. So those are the compounds that we will work on. And those are also the brain areas that we will work on. So the first thing that we did was to see whether these different brain areas, they did have differential contents of these neurotransmitters. And what we did was to separate, to dissect the brain areas. The brain areas were very, very rapidly dissected and frozen in liquid nitrogen. And then the extracts were prepared from these brain areas and deep proteinased to get rid of the proteins, and then put on the HPLC system. So we had the neurotransmitter profile for each brain area. And what we can easily see in this PCA analysis is that these different areas, they are really separated, especially the hypothalamus and the amygdala, whereas the hippocampus and the prefrontal cortex, they overlap quite a bit. So this was the first step that we did several years ago. And this makes us also confident of what we can measure, and that we can go ahead by applying this kind of techniques to different problems, to different kind of problems and conditions. The other technique that we have been using quite a lot is proteomics. And proteomics are a set of techniques that they are focused at the identification of proteins in complex samples. This is also a technique that allows the identification and quantification of these proteins, mostly in a comparative way. So we are comparing two groups to see whether, or to see which proteins are abundant in different proportion in one condition compared to the another one. In proteomics, there are two main types of technologies. One is what is called the gel-based approach. That mostly is where you have your tissue, in our case, the brain areas. So you have to prepare the extract, and you separate all the proteins in this extract in a bidimensional electrophoresis. You then pick and separate the individual protein spots. You digest these proteins with a proteolytic enzyme, usually trypsin, and then you obtain a complex collection of peptides that are injected into the mass spectrometer. The other possibility is to take a gel-free approach. And in this approach, there is no gel, there is no electrophoresis. So what you do is, first of all, directly digest this protein extract with trypsin, usually maybe other proteolytic enzymes, to obtain the collection of peptides, of course, very, very complex collection of peptides, and then separate these peptides by liquid chromatography. And then all these fractions coming from the chromatographic separation and containing the different sets of peptides, they are injected into the mass spectrometer. After a complex data analysis, you can obtain the identification of proteins, the quantification, and usually it's a relative quantification, as I told you. So I'm going to explain now which kind of experiments we have performed to show you a little bit which type of things, of strategies or techniques we can use in experiments related to welfare and stress. So in this case, I would like to present you our work on environmental enrichment in pigs this work has been done in collaboration with Dr. Tony Velarde and his team. Those are ethologists. These are a very recognized group on animal welfare, especially in pigs. So the experiment here was to compare, was quite simple, was to compare two groups of pigs that were raised in two different conditions. One, regular conditions, that means that were the control conditions, the barren conditions. That means a restricted area, surface area. Everything was, of course, within the legal rules in Spain or in Europe, and with a slat floor. And the other group, it was the enriched group, the enriched environment group, that has more space, and they were on a straw floor that allows the pigs to play, to play a little bit with the straw. So both groups of pigs were maintained in those conditions for 4 weeks, and then they were transported to the slaughterhouse. The brain areas were dissected, immediately frozen in liquid nitrogen, and then prepared the extract. And first of all, what we did was to determine the neurotransmitter profile. I have to tell you that this experiment was performed in an experimental farm and also in an experimental slaughterhouse. So that meaning that the animals were really, during transport, the transport was very short because the farm, the distance from the farm to the experimental slaughterhouse is really very, very, very short. And the conditions in the slaughterhouse were very, very mild. Very few animals, only the animals included in the experiment. There were no noise, there were no fights, and also the slaughter was really very careful. The obtention of the brain was very rapid because we had a very experimented slaughterman. Okay, so because when you try to determine neurotransmitters, they have a short life, so it's necessary to do the obtention of the samples, of the brain samples, very, very rapidly, and of course in a very standardized way. So what we obtained in this kind of experiment, well, first of all, I have to say that we also obtained serum at the moment of the slaughter, and what we measured also were some of those conventional serum stress markers that I explained to you in the first time. So in this case, for example, what we measured was the enzyme creatine kinase. This enzyme is a marker of muscular damage. It's a very conventional marker in clinical pathology, used as well in humans, in many species. So what we found comparing the enriched group and the barren group was that the CK activity was higher compared to the enriched one, although they didn't reach the statistical significance, but that indicated, or at least suggested, that the animals in the barren group, they were more likely to have muscular injuries or etc. We measured also lactate. Lactate is the metabolite that is at the end of the anaerobic glycolysis. It's also a marker of stress because when the stressed muscle especially utilizes glucose in an aerobic way, and the last product is lactate that is secreted, secreted into the plasma, so it can be measured there. And here we have again that lactate was higher in the barren group compared to the enriched one, and this is significant. We also measured cortisol, the stress hormone cortisol, in serum, and here we also observed an increase in cortisol in the non-enriched animals versus the enriched environment group. And then we measure also aptoglobin. Aptoglobin is one of these acute phase proteins that I told you before. And again, although the results, they didn't reach statistical significance, we can see that there is a slightly higher concentration of aptoglobin in the non-enriched, in the normal, regularly raised animals compared to the enriched. So that means that using the conventional, very well admit in the scientific community as stress markers, they all indicate that animals in an environmental enrichment are better than in a normal, in a regular environment. So what happens with the neurotransmission? We looked at the neurotransmitter profile in all 5 areas: prefrontal cortex, striatum, hippocampus, hypothalamus, and amygdala. We found some kind of differences in the main metabolites, in the main neurotransmitter, or in some of the metabolites. But I'm showing you the most clear difference between both groups that we found in the striatum and referring to the actual neurotransmitters, so dopamine or serotonin. And what we saw is that dopamine as well as serotonin, the levels were higher in the striatum, in the enriched group, compared to the barren group. And our conclusion is that pigs in this barren condition may feel less reward, taking into account the role, especially of dopamine in the striatum. Dopamine in the striatum has been very, very much studied in humans related to several kinds of addictions. Okay, so then we wanted to do a proteomic analysis, and we choose the hippocampus. And we choose the hippocampus for several reasons. First of all, it's involved in very important functions of the brain that are related also in stress, but also in other capabilities of the pig brain. And also because another reason is that the hippocampus is very easily dissected and it's very easily recognized, and it's very well separated from other brain areas. The name of hippocampus comes because of it resembles the seahorse shape, and the morphology and also the development of the hippocampus is very well known. So this is the draw from Camillo Golgi that is many years ago. And he already recognized like 2 main regions in the hippocampus, the gyrus dentatus, which is this one, and the other one is what is called the cornua ammonis, the almond horn, that is this one, and it's divided in at least 3 areas, CA1, CA2, and CA3. So because the important role of the hippocampus and also the easy dissection and the very well-known morphological characteristics, then we choose the hippocampus to perform the proteomic analysis. In this case, we choose a GLF3 approach that it's called the technique is called ITRAC, for isobaric tag for relative and absolute quantitation. And I will explain you very simply this technique. You have samples from different animals, and what you do is you have the original protein extract. Okay, so you have to first to treat the proteins, to avoid the formation of covalent bonds between the different proteins. And then you trypsinize, so you degrade the protein into peptides, and then you label this set, each set of peptides with a different tag. Okay, so we have these different tags or different sets of peptides that are labeled in a specific way. The way to set up the experiment is that in our case, what we had was 20 individuals in the barren conditions, 20 hippocampus, 20 hippocampus from individuals in enriched conditions, and then 8 control samples. Control samples mean that it's a pool of all samples, because in these control samples, what you have to have is to be sure that you have all the proteins that can be identified in any of the samples that you are working with. So we have 48 samples, so we had to do 6 ITRAC labeling reactions, because it's 6 per 8, 48 different labelings, okay? So after the labeling, you combine all the peptides and you put it on the mass spec. In this case, it was a mass spec, this Orbitrap mass spec. And then the system can identify all the reporter ions, and using the corresponding adequate databases that for swine is complete, therefore sus scrofa is complete, I don't know about poultry, and then you can quantify in a comparative way, the comparing groups, the protein composition of each of the groups. So what we afterwards, well, we identified more than 15,000 peptides that corresponded to almost 2,500 proteins, but only 59 of these 2,500 proteins were found differentially abundant proteins between the barren and the enriched groups. Using a criterion not very strict, that was a 1.3-fold of difference between one group or another, up or down, and with a significance of P005 as usual. So this gene ontology analysis using Panther, this software, this platform, allows you to characterize in a broad manner the different types of proteins. You can check or you can discriminate by molecular function, by biological process, or by cellular compartment. So in this case, I'm showing you the molecular function. As you can see, most of the proteins, of the 59 differentially abundant proteins, correspond to structural proteins, and 31% corresponding to proteins with catalytic activity. That means enzymes. So what we did afterwards was, of course, to identify all those proteins. And what we saw about the structural proteins was that they correspond to 2 types of proteins. The majority of them, 68%, were ribosomal proteins. You can see here in the list that there are ribosomal proteins coming from the small subunit of the ribosome, or from the large subunit of the ribosome, and most of them were ribosomal proteins from the structure of the ribosome, but there were also 2 proteins, enzymes in this case, that are also involved in translation. Those are tRNA ligase, that means the enzymes that bind the amino acid to their correspondent transference RNA. And all those, except one, were overexpressed, overabundant in the environmentally enriched group versus the barren group. And the other 32% of these structural proteins were related to the cytoskeleton. And you can see that most of them are tubulins. Tubulins are the proteins that form the microtubules of, for example, the axons of the neurons. So here we have two types of proteins, very, very well defined. We also looked at the enzymes, the proteins with catalytic activity. And here there is a little bit more of diversity. But what we found is that we had also a set of proteins that they are all overexpressed in the environmentally enriched group, and that they are related to the respiratory chain, to the mitochondrial respiratory chain. So with another network analysis using another kind of tool that is called string. And with the string, or what the string does is to cluster all proteins that are interconnected in a way or another. So, and here we have very clearly a cluster containing the ribosomal or corresponding to the ribosomal proteins. We have also another cluster that corresponds to the structural proteins, the tubulins, and we have another cluster that is related to these mitochondrial proteins. We analyze also the data using another tool that is called Reactome. And with Reactome, what Reactome shows you is you upload your list of proteins. And what Reactome does was to analyze in which kind of pathways are these proteins, or do these proteins participate. And here again, we found that mostly the proteins that we have identified as differentially expressed are involved in translation and protein folding, and also in microtubule-dependent trafficking. There is also a connection with the retrograde transport, that is the transport that occurs in the axonal neurons from the synapses to the core of the neuron. So everything was conversion, fitting together in the same kind of a scenario. No? So the conclusions of this work are that PECs in barrier conditions shows a higher degree of stress. PECs in barrier condition feels less reward. We'll see. And that this enriched hippocampus potentially has increased capacity for protein synthesis, increased capacity for axonal dendrite transport, and increased oxidative phosphorylation. That means capacity to produce ATP. So I'm going to show you now the second experiment that is about intrauterine growth restriction. And this condition provokes the birth of low birth weight offspring. No? Here we have two brothers, not two siblings, coming from the same mother. One is a normal weight piglet, and the other one is one of these IUGR piglets. And this condition appears when there is a deficit, a nutritional deficit, also in some placentary conditions, especially in women, that restrain or limit the availability of nutrients and oxygen to the fetus. So that leads to what is called an asymmetrical fetal development, because it produces an effect that is called brain sparing. That means that the brain is preserved, and the size of the brain is almost normal when you compare the heads of the two animals. And on the other side, other organs, as for example the liver, they grow much less. Even though to have this brain sparing fact doesn't mean that the brain develops correctly. We have used a porcine model for UGR, that is the nutritional restriction of the mother during the last 2/3 of pregnancy. And in the advantage of this model is mainly that UGR and normal piglets, they are born from the same mother. So we discard one very important effect, that is the mother factor. So this work was made in collaboration with Dr. Antonio González Bulnes, that is in the INIA in Madrid. So intrauterine growth restriction is also a condition that affects humans. And in humans, it's very, very interesting because although the fetuses, they are born with low birth weight, and they may have another complications, when these babies, they become adults, they are not low weight. On the contrary, they develop obesity. And not only obesity, but also diseases that are derived from obesity, that as, for example, type 2 diabetes and cardiovascular problems. And they do have also increased presence of brain problems, brain disorders. This is what is called the thrifty phenotype. And the pigs, well, we are using a special breed of pigs, that is the Iberian pig, that it shows this kind of thrifty phenotype. So it was already in the literature the possibility of try to prevent this condition using antioxidants in the maternal diet. So this is what we tried in pigs. So the first objective was to analyze the effects of supplementation of the maternal diet with hydroxytyrosol, which is an antioxidant, okay? And see what happens to the central nervous system, the brain of the offspring. And this was the experimental design. Those are we had at the beginning 51 pregnant souls. They were calorically restricted after the first third of the gestation. And then these animals, we obtained the brains from 100 days old fetuses, 1 month piglets, and 6 months pigs. And we have 2 groups of mothers, the control and the supplemented, the mothers that were supplemented with HTX. So what we found with the neurotransmitter was very peculiar. This is the neurotransmitter profile that we found in amygdala, in hippocampus, and the prefrontal cortex from fetuses in red, in 1 month piglets in green yellow, and in 6 month pigs in blue. And as you can see, in fetuses, in 100 days fetuses, there are a lot of differences, no? In all the neurotransmitters and metabolites, okay, and in all the 3 areas. Usually, the concentration of the neurotransmitter, for example, dopamine or serotonin, is higher in the treated mothers than in the control mothers. But these differences between the 2 groups, they disappear after the pigs are born. So the conclusion of this part of the experiment is that the effects of the supplementation of the maternal diet with HTX are only visible in 100 days fetuses, but not postnatally. And then what it can be concluded is that the continuous presence of HTX is needed to maintain its effects on the brain. What we did afterwards was to look at the morphology of the hippocampus. This is the gyrus dentatus, and this is the horn, ammonin horn, okay, CA1, the ADA CA1. And we stained our preparations with 2 markers. One is N, that is a marker for mature neurons, and the other one is double cortin, that is a marker of immature neurons. I'm showing only the staining with the N, which is nicer, and it can be seen very clearly, the results. What we can see here is that in 100 days of fetuses, those are the control, the piglets, or the fetuses coming from control mothers. And these are the fetuses coming from HTX treated mothers. And what we can see is that here we have, in the treated models, we have much more mature neurons than in the control, and the pigs from control models, in the piglets and the fetuses from control models. That happens in CA1, and also, maybe not as visible, in the gyrus dentalis. But again, these differences that we can see in fetuses, they are not observed in postnatally or in pigs postnatally, nor in 1 month old pigs, nor in 6 month old pigs. So the idea is the same. No? The fetuses in the models treated with the antioxidant show a higher number of mature neural cells and lower in mature neural cells. That indicates that HTX in the model induces a faster neuron differentiation process in the fetus. And this is only seen in the fetuses, but not in, it's not seen postnatal. So it's also a proteomic analysis. We're using this time another kind of labeling that is called TMT, tandem mass tagging. It's similar to the ITRA. So finally, what we are doing is to compare the protein composition of both groups, okay, control and HTX. And we found only 11 differentially abundant proteins. 3 more abundant in the piglets coming from treated models, and 8 more abundant in the piglets treated with untreated models. Okay, and these proteins were quite peculiar because the 3 more abundant proteins in the fetuses coming from models treated with the antioxidant or supplemented with the antioxidant, those were enzymes that are involved in detoxification, or are antioxidants, or they involved in neuronal development again. Whereas the 8 more abundant in the fetuses coming from control models, they are related mostly to ribosome, and also there is one that is related to lipid metabolism. So this is the conclusion. Upregulated proteins in the HTX group. Remember that those are piglets, fetuses coming from supplemented models, and the control are fetuses coming from unsupplemented models. So those are involved in detoxification, cell protection, and synapses formation, suggesting a potential mechanism for the neuroprotective effects of hydroxytyrosol. And what happens with intrauterine growth restriction? Since the effects of hydroxytyrosol were only visible in 100-day fetuses, so we looked only at the fetuses when comparing piglets or fetuses with normal weight and low weight. And what we saw is that here are the results of the histology. What we did here was to count the cells in the CA1 layer, the mean size, well, more or less, those are parameters that are related. And what we can see is that if you look at the control, for example, okay, the fetuses with normal weight has more cells, but smaller cells than the low birth weight in the control group. Okay, so being an IUGR fetus, that means that the number of cells in CA1, in this part of the hippocampus, was lower. Okay, what happens in the hippocampus of fetuses coming from supplemented models supplemented with the antioxidant hydroxytyrosol? This difference disappears. So the number of cells in fetuses with normal weight and low weight is the same. And with the gyrus dentatus is more or less the same, but it's less evident. So the idea is that the differences observed between normal and low birth weight groups in the control group disappear when you compare groups whose mothers are treated or have been treated with the antioxidant. So at least looking at these results, no, supplementation of the mother's diet with HTX is able to reverse the effects of IUGR. And what happens with neurotransmission? We looked also all the neurotransmitter profiles, but there was almost no difference between both groups, normal and low birth weight. But there was one difference that was, this is serotonin, okay? And we can see that in serotonin, if we compare, these are normal, the dark ones are normal weight fetuses. That means that the treatment of the mother with hydroxytryptamine, with a hydroxytyrosol, increases. These differences also disappear in fetuses whose mothers have been supplemented with HTX. Again, the differences observed between normal and low birth fetuses, weight fetuses, in the control group disappear in the supplemented group. So those are the conclusions, but as I already checked, maybe it's already time to finish. So the general conclusion is that the differences observed between the control group, between normal and low weight fetuses, disappear when you compare the treated groups. That meaning that supplementation of the mother's diet with this antioxidant, hydroxytyrosol, is able to reverse the effects of IUGR. Of course, those are only like descriptive results. We don't know whether these piglets, they perform better when they are born or not. But at this molecular level, the effect is clear. I have to say that hydroxytyrosol is an antioxidant that is a natural antioxidant. And it's very, for example, olive oil is very rich, especially virgin olive oil is very rich in hydroxytyrosol. I have the third experiment, but I think that we are running short of time. So there is not much neurobiology in it. So I'm going to skip this thing. And those are the general conclusions of the work that I have presented to you. Stress and welfare can be approached with traditional techniques, quantifying these biomarkers like stress hormones, acute phase proteins in serum samples. Other samples, for example, cortisol or corticosterone can be determined or quantified in hair. They can be also determined in feather. They can also determine it in saliva, in feces. So it's a quite interesting way because cortisol measured in plasma or serum is an indicator of acute stress, but measured in other samples, like, for example, hair or feathers, that they accumulate this cortisol, it's an indicator of chronic stress. So you can differentiate between acute stress and chronic stress by using, by determining cortisol or cortisol derivatives in different types of samples. We also can conclude that the neurotransmitter profile can detect changes caused by environmental conditions, and also that these proteomic approaches are useful to provide new perspective and interpretation of stressful conditions. And I would like to finish by recognizing the work of my three former PhD students, Laura, Dani, and Natalia, who performed all these works. This is my town. This is about 25 kilometers from Barcelona. And this is one of the holidays that where the animals are blessed. And it's a very, very nice holiday. And I want also to thank you, the people in the service of the veterinary clinical biochemistry that have been here with me for a long time. Also the people from the Institute of Neuroscience that helped us, especially at the beginning, with all these neurotransmitter studies. And also Tony Velarde and Antonio González Bulnes, who helped us with all the animals experiments. Martí Pomarola, which is the neuropathologist in our faculty, and also the people from the proteomic platforms that really, really helped us a lot to interpret this kind of experiments. So thanks to you for your attention.